Welcome to the second video on the unit on microbial pathogenesis. In this video, we're going to talk about the first three steps in the mechanism of pathogenesis. What that means is, what does a microbe have to do in the body to actually be a pathogen? There's 5 total steps, so we'll cover the first 3 in this video and then the last 2 in the next video. Let's go ahead and get started. So here are the 5 overall steps for pathogenesis. What that means is, to be a pathogen, a microbe has to be able to do these 5 things. So the first is there has to be some sort of exposure to the pathogen and an entry into the body. The second is attachment to tissues and colonization. So the microbe has to attach and then start to grow and divide. The third is immune avoidance. So in order to be able to colonize and grow, the microbe has to be able to avoid the immune system, because remember, we just learned about all the different ways we have in the body to recognize foreign things coming in and destroy them. In order to not be destroyed, the microbe has to have ways to avoid that. The fourth step is actually causing the infection, which is damaging host tissues. And then the last step is going to be to exit the body and to undergo transmission if this is a communicable infectious disease, which will be the ones we're mainly focusing on, those ones that can be contagious between different individuals. So the first step in microbial pathogenesis is entry into the body, which means you have to be exposed to a pathogen. So where do you actually get these exposures from? The normal host, or the normal habitat for a microbe, is considered the microbial reservoir, so where it normally lives until it enters your body and becomes a pathogen. There are 3 main types of reservoirs. The first are humans in general, so those can be people who are sick and they're contagious and they're spreading the pathogen to other people, or they could be asymptomatic carriers who actually aren't sick, they don't know they have disease, but they still have the microbe in them and they are spreading it. That's we're having a large conversation about that with COVID-19, as we were seeing what is likely a higher percentage of asymptomatic carriers spreading disease than typically happens for most pathogens. The second type of reservoir are animals. So there are some pathogens that can jump from animals into human beings. When they make that jump, they're called zoonotic pathogens. So zoonotic pathogens are ones that are able to jump between animal species and jump into humans. We think that COVID-19 first arrived in human beings as a zoonotic infection, jumping from other animals. And then the third reservoir is an environmental source. So there are some microbes that aren't actually trying to live in your body. Right? They live in water or they live in soil, and that's their natural habitat. And it just so happens that you came into contact with where they normally live, and they've gotten inside the human body somehow. So coming into contact with a pathogen doesn't do anything if that pathogen can't actually enter the body. So the main sites of entry, or the portals of entry, are the sites that are in contact with the environment. So typically you're going to have a pathogen enter through some sort of break in your skin. They're going to enter through the mucous membranes, and so either through fecal-oral routes, which are through the gastrointestinal tract, respiratory routes, which are breathing into the mucous membranes of the respiratory system, or urogenital tract through the genital mucosal membranes. We also have what's called peritoneal entry, which is when you have an injection in, so either through things like injection drug use, or we see this with things like mosquitoes or ticks that are breaking through the skin by injecting into the skin themselves. Typically, the portals of entry are also tied to the portals of exit, so we'll spin back around to these at the end when we get to that step 5. Once a pathogen has entered the human body, the next step, it has to actually adhere to the host cells. This is going to prevent the pathogen removal. So remember, you have lots of mechanical ways to remove microbes in the innate immune system, whether that's traveling through the gastrointestinal tract and out the other end, whether that's the mucosal cilia beating up to make that pathogen come back out, say when you cough or sneeze. In order to actually colonize the body, the microbes need to stick to our cells and tissues. And so they have microbial adhesins, and we can see an example of those here. Remember, we talked about the role of pili in bacteria for adhering, for sticking to stuff. Here we've got a pili with an adhesin protein interacting with a specific host cell glycoprotein. We talked about adhesion when we talked about viral replication, because all the viruses had to adhere to our cells to be able to enter the cytoplasm. All of your pathogens are going to have to do this, but they don't all have to get into the cell the way viruses do. So all pathogens need to have some sort of adhesin molecule that's going to interact with some sort of receptor on human cells and tissues so that the pathogen doesn't get flushed out of the human body. Just like in viruses, this adhesion, this interaction of the receptor and the specific protein on the pathogen is going to dictate the host range. So the pathogen would be able to bind to any species that has that receptor. And the tropism, it'll be able to interact with any tissue type that has that receptor. We can see some of the bacterial adhesions in this table here. So Streptococcus pyogenes, which is the causative agent of strep throat, uses an adhesion called protein F to bind to the respiratory cells. That gives us its tropism. Whereas something like Vibrio cholera uses a pili to bind to the epithelial cells because it's a gastrointestinal disease. So again, we see the tropism tied to what the adhesin protein is and what receptor that protein is going to interact with. For bacterial pathogens, there's two main types of adhesins. The pili, which we talked about when we first talked about bacterial structures, are cylindrical structures made up of pilin protein, but at the tip they have a different protein. So at the very tip they have an adhesin protein. You can see it here in the picture. Notice it looks different than the rest of the pili itself. This is because it's not building the pilus itself, it's what's going to adhere to a receptor. Different pili have different adhesin proteins, which allow them to bind to different things. But no matter the type of adhesin, most of those are going to be binding to cell carbohydrates. You've got type 1 pili, which is here that I just highlighted. These tend to be very static, so they're just sticking out there waiting to bump into a receptor. And you also have type 4 pili, which actually will grow and contract and move around. So you can see this one here was extended, and then it kind of comes back in. You can see this one has been shrinking, and so these will shoot out and come back looking for something to bind to. Bacteria also have non-pilus adhesins. These are typically microbial proteins that are found on the surface of the cells, and they're going to bind to other host cell proteins. So if you're binding to a carbohydrate, the pathogen is typically using a pili. If it's binding to a host cell protein, it's typically using a protein of its own. Remember, we also talked about biofilms that were able to strongly adhere to surfaces. So these were these high-density cell communities that were embedded in that exopolymer that made them sticky and made them difficult to remove. They're able to adhere to both living and non-living surfaces, and so we find biofilms really important in infectious diseases for chronic infections, ones that don't follow the little acute pattern where the disease goes away, but that the infection lasts long-term, because those biofilms are hard to get rid of. So this is things like dental plaque in the mouth. We also see these form in the lungs and in the bladder. So for people that get recurrent urinary tract infections, oftentimes it's because they have a biofilm on the inside of the bladder that continually sloughs off planktonic cells into the bladder itself. They grow and replicate and cause a UTI. When you take antibiotics, it kills all those free-floating cells in the bladder, but doesn't destroy the biofilm from the wall itself. And so the next week, some more slough off, and you end up with another UTI. Just adhering to the cells or the tissue is not enough to actually cause disease. So once a pathogen gets into the body and sticks, it then needs to grow and divide and make more organisms. So if this is a viral pathogen, it has to get into the cells and use them to produce more viruses through viral replication. This is a bacterial pathogen, it goes through binary fission. If it's, say, a fungi, it's going to be dividing with spores inside the body. And so we have to make more of the organisms in order to actually damage tissue, which is what leads to disease itself. This colonization is also going to require evasion of the host immune response, because remember, the immune system's there to destroy these microbes as they get in. In order to grow and divide and make more of themselves, the pathogens have to first evade those immune responses. Anything that a microbe produces that allows it to avoid these host defenses is considered a virulence factor. So remember, virulence factors are just anything that the microbe produces that allows it to be a pathogen. So those adhesins we were talking about, those are all virulence factors because you have to adhere in order to be a pathogen. Anything that's produced that allows us to bypass the host defenses is going to be a virulence factor. And often these things are produced once the pathogen is in the body, but not when it was necessarily back in its other reservoir, especially if that reservoir was environmental. This is why we care about studying things like gene expression, because once in the body, you can identify what's now being produced by the pathogen and start to find some of these virulence factors. The three main strategies that pathogens can use to bypass the immune system and the host defenses are: one, hiding. So if they're intracellular pathogens and they get in the cell themselves, they can avoid some of the immune system responses. Two, disguising themselves so they don't necessarily look the way a pathogen would. Or three, attacking the immune system before the immune system can attack it. We're going to look at each one of them individually. One way to avoid the immune system for a time is to be an intracellular pathogen. Some pathogens are obligate intracellular pathogens, like viruses, where they have to live inside another cell, but others are facultative intracellular pathogens, where they'll go inside a cell to hide for a little bit, but then they can grow outside the cell just fine. They're going to have to get in through either endocytosis or phagocytosis, and both of those pathways end with fusion of the lysosome. And so these intracellular pathogens, if they're hiding inside a cell to avoid destruction, they have to be able to prevent being killed by the lysosome, or they've defeated the purpose. There's three ways this can happen. There are some pathogens that actually escape from the phagosome. So after they're phagocytosed, they destroy the vesicle around them, and then they grow very happily in the cytoplasm. We see an example of that here with Shigella. So this is a fluorescent micrograph, and you can see the red Shigella bacteria. And these are really cool intracellular pathogens because they actually polymerize our host actin, which is what you see this comet tail is. And they use that actin to push them around the cells. So if you watch a video of these, they actually fly around the cytoplasm. And it's one way they transfer from cell to cell, is the actin will actually push them out of the cell and into the next one without actually ever having to leave that cytoplasm. This allows them to avoid things like phagocytosis by macrophages that might be out in that tissue looking to chew them up. There are some microbes that will inhibit phagosome-lysosome fusion. So they go in through the phagosome, and then they have proteins that prevent fusion of the phagosome with the lysosome. Whereas the lysosome that has all the killing enzymes, so those pathogens grow perfectly happily inside the phagosome itself. And then there's a third, much smaller group of pathogens that just say they don't care about all of your destructive pathways and they can grow perfectly fine in the phagolysosome. They have to have virulence factors that allow them to detoxify that lysosome so that they don't get killed. While intracellular pathogens can hide from the immune system by living inside the cytoplasm of cells, extracellular pathogens have to have other ways to avoid the immune clearance. The two main ways they do this is to disguise themselves so they don't look foreign, or to attack. Having a capsule is one of the most common ways to disguise yourself. Remember, capsules are this polysaccharide layer that surrounds the cell. We can see it here. And what this does is it disguises any of the antigens that would have said, hey, I'm a foreign cell, because they're buried under this sugar layer. And remember, we talked about polysaccharides are not very immunogenic. And so typically the capsule won't set off an immune system response. It doesn't necessarily look foreign, and it's hiding those antigens that the antibody or the phagocytic cells would have recognized. Other ways pathogens can kind of disguise themselves is to constantly mutate the proteins on their surface, so those antigens that the immune system is looking for. If it's mutated and it looks slightly different, the antibodies might no longer bind to it. This is what influenza does with antigenic shift and antigenic drift, and it's why we need a new influenza vaccine every year, because the flu virus from last year has mutated and looks a little bit differently this year, which means it has disguised itself from the immune system. The other way that bacteria can avoid the immune system is to just attack the immune system back. And so some of these produce proteins that mimic cytokines. So whereas the immune system might be saying we need a Th1 response, the pathogen says, no, 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 you need a Th2 response, and then the immune system does something wrong. Or we can have enzymes like the protease seen here that degrade things like antibodies. So the immune system has mounted a response, and then the bacteria fights back and chews that response up. They can also do things like trigger apoptosis of phagocytic cells, so they kill the phagocytic cells before the phagocytic cells can kill them, or they can infect and destroy the lymphocytes to prevent an adaptive immune response.